So ladies and gentlemen, you are all welcome to my YouTube channel. Today we are going to look at the biology. Specifically, we are going to look at DNA as a topic. So what is a DNA? The DNA stands for deoxyribonucleic acid. It stands for deoxyribonucleic acids. And this DNA is found in the nucleus in plant cell. The DNA is found in the nucleus, and the DNA that is found in the nucleus is called nuclear DNA. It's called nuclear DNA. And it is also found in the mitochondria, And the DNA that is found in the mitochondria is called mitochondrial DNA. Mitochondrial DNA. So DNA stands for deoxyribonucleic acids. It is found in the nucleus of the cell. In the case of animal cell, it is also found in the mitochondria in the case of plant and animal. Also, we have chloroplasts in plant. Chloroplasts. So chloroplasts also contain DNA. So the chloroplasts also contain DNA. So it means that both nucleus and the mitochondria contain DNA in the case of animal cell. But in the case of plant cell, because plant cells have chloroplasts in addition to nucleus, mitochondria, and chloroplast. So it means that we have, in the case of plant cell, we have DNA in the chloroplast, we have DNA in the mitochondria, and we have DNA in the nucleus. And in addition, the concentration of the DNA in the nucleus is high, is very high compared to other cells. So it means that the amount of DNA that are found in the mitochondria and the chloroplast is very small. So the concentration is small. So therefore, most of the DNA are found in the nucleus. And one very important information that we should understand that each and every cell in living organism contain DNA. Each and every living cell of living organism contain DNA. But now the question is, is the DNA the same for all the cells? That in our liver cells, brain cells, kidney cells, pancreas cells, kidney cells, are they actually have the same DNA? Is the DNA the same for all these organs, for all the cells that we have in the body? And we should understand that, like, for example, an average human being have about 7.2 trillion cells. These are the number of cells that are found in average human beings. And we have different cell types. So these are the number of cells that are found in average human beings. And we have over 220 different cell types. We have over 220 different cell types, ranging from myocytes, that is the cells of the muscles, hepatocytes, the cells of the liver, erythrocytes, the cells of the red blood cell, neurocytes, nephrocytes. All these are different cells that are found in human beings. So, but now the question is the DNA of the myocyte, hepatocytes, and the nephrocyte the same? Are they the same? The answer is yes. Nearly each and every cells of every living organism contain DNA, and those DNA are the same across all different cell types. But you should understand that because there is what they call a differential gene expression. What is gene expression? When you say gene expression, what does that mean? A gene expression is always, we look at it always from the point of central dogma of molecular biology. So gene expression is when you have DNA, converted into RNA, and then the RNA will be converted into protein. So this is what we call a gene expression pathway, or we call it central dogma of molecular biology. DNA converted into RNA through a process called transcription. It is called transcription. And the RNA is converted to RNA copy itself into RNA through the process called translation. So this is what we call the central dogma of molecular biology, although the DNA at the same time can also copy itself. The DNA can also copy itself. So when DNA copy itself, the process is called replication. So when DNA copy itself into another DNA molecule, the process is called replication. So now what we are saying is all the DNA, because one very important thing that we should understand, that DNA is the language of life. DNA is the language of life. If we say DNA is the language of life, what does that mean? It means that the way you are, the way you appear, the way you interact, the way you behave is all because of your DNA. Like, for example, in the DNA we have a genes. Like in human beings, we have about 35,000 genes. And each and every genes have the ability or have the potential to produce a particular protein. Like, for example, out of the 25,000 genes that we have in human, we have a gene that can produce melanin. What is the melanin? Melanin is a protein that contributes to our color of skin. So the more the amount of the melanin produced, the darker the color you have. And the low the amount of the melanin you produce in your body through this concept. Because as we said, in a DNA is where we have these 25,000 genes. And like, for example, in the DNA we have a genes that can produce melanin. So the genes that can produce melanin will be converted into melanin RNA. It will be converted into melanin RNA, and the process is called transcription. And the melanin RNA will also translate itself into melanin protein. So melanin is a protein. So through the process called translation. So this is how proteins are produced in the body, and proteins in our body is actually what determine each and every physiological activities that are taking place in our body. So therefore, the central dogma of molecular biology is a process that involves gene expression. So DNA contains about 25,000 genes, and each and every gene in human being has the potential to produce a particular proteins. And that proteins that produce is what actually determine the activity of a particular cell. Like, for example, we have red blood cell. In a red blood cells, we have a protein called hemoglobin. So therefore, in our red blood cells, we have a DNA, and the DNA in the red blood cells have the ability to produce hemoglobin, and that is what makes the red blood cells to be involved in transporting oxygen into the body tissue or throughout the body tissue. And at the same time, we use brain for cognitive function, for educational purposes, for a lot of activities like calculation, memorization, intellectual capacity. We all use our brain. And it's because in the brain there is a DNA that expresses a protein that make the brain to produce a protein that make the brain to carry out all the cognitive function, memorization, calculation, and so many other things. And it is because the DNA is responsible for controlling each and every character. So therefore, if in your body you have a DNA that produces a protein that will make you intelligent, so it means that you can inherit that DNA from your father or from your mother. So DNA can be inherited from our parent. So if our DNA have a genes that produce a lot of proteins that are associated with the cognitive or intellectual functions, then you can inherit that and you can inherit the intelligence. So DNA is a language of life, and we are the way we are is because of our DNA, and the way we have our color of skin is because of our DNA, and we are intelligent because of our DNA. We talk a lot because of our DNA, because in our DNA we have genes called FOXP2, and that is what actually makes us to talk. So the more you produce the FOXP2, then the more you talk. So this is what we call central dogma of molecular biology. So DNA is very, very important. So, but now, what we are saying is this DNA is same across all the 200 different cell types. The answer is yes, they are the same. So, but what because since it is a DNA that dictates, it's a DNA that will dictate the function of a cell. It's a DNA that will dictate the function of a particular cell. So, but now the question is, someone will ask, why is it that the functions of the brain cells is different from the functions of the liver cell? And the functions of the liver cells is different from the functions of the kidney cell. The functions of the kidney cell is also different from the functions of the pancreas. So why is it that there is this difference? It is because different proteins are expressed or are produced at different cells, but from the same DNA, and that is what we call differential gene expression, which leads to what we call cellular differentiation and specialization. So, but now, how is this the stem cell originated? So we know that actually during fertilization, we have a sperm cell plus egg cell plus egg cell. So sperm cell and egg cell join together. A sperm cell and egg cell join themselves together to produce what we call a zygote. To produce a single cell called zygote. And this zygote is a single cell. This zygote is a single cell, and it has the potential to differentiate, so it undergo mitosis. Like for example, this is fertilization. So when you have egg and sperm cell, a zygote is produced through fertilization. Of course, if you could remember, fertilization takes place during meiosis. So fertilization takes place during meiosis. meiosis. So after you have the fertilization, you have zygote. So this zygote will now further divide itself to produce from, you have sperm cell and egg cell joined together to produce a zygote cell. And this zygote cell will also further divide itself through the process called mitosis. So during the mitosis, one zygote cell divides into 2, and 2 divide into 4, 4 divide into 8, and 8 divide into 16. So after, and remember, one of the feature of mitosis is that the daughter cell have the same genetic material as the parent cell. So it means that the DNA in the parent cell and the DNA in the daughter cell are the same. So after you have a number, maybe 1,000 number of a zygote cell, 1,000 number of a zygote cell, 1,000 number of zygote cells, they will start differentiating. And that is what leads to the cellular differentiation and specialization. Like for example, let's say you have thousands of the zygote cells. And some of these zygote cells will be directed to differentiate to produce red blood cell. So if maybe let's say 100 or let's say 50 of the zygote cells are directed to produce red blood cells. So what will happen? It means that in these 50 cells, the gene that is responsible for producing hemoglobin, that is responsible for producing hemoglobin, will be expressed. That is, it will be produced in the red blood cells, and that is why you have hemoglobin in the red blood cell. And at the same time, some of the cells, maybe another, let's say 100 cells, will be directed to produce pancreas. So what happened? You know that in the pancreas we have two major proteins that are found in the pancreas. We have insulin, glucagon, and somatotropin. They are hormones, and they are all found, or they are all produced in the pancreas. So it means that in these 100 cells that are directed to produce zygote cells, it means that the DNA of these 100 cells will be also instructed to produce or to express the gene of insulin, glucagon, and somatotropin. And these ones, they are hormones, and at the same time, they are proteins. And this is what will make these 100 cells to be differentiated into pancreas cells. But the DNA that is found in these 50 cells that are instructed to produce red blood cells is the same. And with these 100 cells that are instructed to produce pancreas cells, the DNA, they contain the same DNA, they contain the same genes, but only that some genes are directed to be expressed in red blood cells, while those genes are silenced in the pancreatic cells, but they contain the same DNA. So ladies and gentlemen, we should understand that our cells contain the same DNA. So each and every cell in our body contain the same DNA, and each and every DNA has the potential to differentiate or to express a different protein or a different genes. So this is what you call differential gene expression. So differential gene expression is what makes one cell differ from one another. Like for example, in the pancreas, we have a gene for producing insulin, we have a gene for producing glucagon, we have a gene that can produce hemoglobin, we have a genes that can produce phosphatase protein, but it's only that the genes that are producing hemoglobin and phosphatase will be silenced. They will be silenced, and only those genes that are responsible for producing insulin, they are the one that will be expressed in the pancreas. And this is what we call cellular differentiation and specialization. But we have the same DNA, we have the same genetic material. So as we are doing the lecture, we are going to look at each and every of these steps to replication, transcription, and translation. So now, what constitutes DNA? So the DNA is made up of basically 4 letters. 4 letters is what make the DNA. And those letters are letter A, letter G, letter C, and letter T. These are the letters, we call them nucleotides. We call them nucleotides. These are the 4 letters that make up the DNA. So DNA is like a book that is written with 4 letters in different permutation and combination. And these letters are A, G, C, T. There are A, G, C, T, and we call them nucleotides. So DNA is a polymeric molecule. DNA is a polymeric molecule. So you can say it's a biopolymer. It's a biopolymer. It's a biopolymer of nucleotides. It's a biopolymer of nucleotides. So DNA is a biopolymer of nucleotides. So when you say DNA is a biopolymer of a nucleotide, it means that the building blocks of DNA is nucleotides. So it means that thousands and millions of nucleotides will join together to produce what we call DNA. So these are the thousands of nucleotides that join together to make what we call a phosphodiester bond to produce DNA. So DNA is made up of thousands of nucleotides that joins together through phosphodiester. So that's why we say DNA is a biopolymer of nucleotides. And what are these nucleotides? What are the composition of these nucleotides? The nucleotide is made up of 3 major components. Nucleotide is made up of 3 major components. It's made up of number 1, nitrogenous. Is made up of nitrogenous bases, that is number 1, and it is made up of sugar, and then finally it's made up of phosphates. It is made up of phosphates. So now we are talking about nitrogenous base. So nitrogenous base is divided into 2. It's divided into 2. We have pyrimidines and we have purines. And these purines and pyrimidines and purines, we have them like this. So purines, example of purines is adenine and guanine. Adenine and guanine and adenine are the purines, while pyrimidines, we have three of the pyrimidines. We have cytosine, we have adenine, and then finally we have thymine. So these are the purines. So we have purines. So purines, we have adenine and guanine, and pyrimidines, we have three. We have cytosine. Now this one is not adenine. We have cytosine, we have thymine, and then we have uracil. So these are the three nucleotides that we have. So thymine and cytosine, they are all found in DNA. This one is also found in DNA. But in terms of this uracil is only found in RNA. So it means that for the DNA, there are 4 letters that form or that constitute the DNA. We have cytosine, thymine, adenine, and guanine. So that is why these are the 4 letters that we have that make up the DNA. And then we have sugar. The sugar that is found in the DNA is pentose sugar. It's pentose sugar. When you say pentose sugar, it means it's a 5-carbon sugar. It is a 5-carbon sugar. And the name of the sugar that is found in the DNA is deoxyribose. It's deoxyribose sugar. It's deoxyribose sugar. So it means that the sugar that is found in the DNA is called deoxyribose sugar, and the nucleotides, the nitrogenous bases that also found in the DNA is cytosine, thymine, adenine, and guanine. Then finally, phosphates. We have phosphates. So apart from the nucleotides, we also have what we call nucleosides. What is the difference between nucleotide and nucleosides? So a nucleoside is only when you have nitrogenous base. When nitrogenous base is when you have nitrogenous base plus the pentose sugar. So nitrogenous base and the pentose sugar will give you nucleosides. And then nucleosides plus phosphates will give you nucleotide. So what we have in the DNA, is it nucleoside or nucleotide? Is nucleotide that we have in the DNA. Okay. If these nucleotides, we call them sometimes this nucleotide you can call them bases. So in human beings there are about 3 billion base pairs. So 3 billion base pairs mean 3 billion nucleotides. So 3 billion nucleotides are found in the DNA. That is 3 times 10 raised to the power of 9 base pairs. So it means that 3 times 10 raised to the power of 9 base pairs are the number of the base pairs that are found in human DNA. And 99% of the human DNA is the same. They are the cell. 99% of my DNA, your DNA, they are the cell. And it's only that 1% difference that makes you different from the way I am. So human being, we have 3 billion base pairs. We have 99% similar DNA. But what makes you and I differ from one another is because the DNA have only 1% difference between mine and yours. And that is why you are the way you are and I'm the way I am. So it's just these differences that we have across individual or across humans. That is what make each and every one of us different. So in human beings, we should understand that in human beings, there are fundamental things that make us to behave the way we behave, to react, to respond to different disease conditions, to resist different disease conditions, to respond to drugs, to be intelligent, to talk the way we talk, to be smart. All is because of this DNA. So DNA is the language of life. And we shouldn't actually take it for granted, because without understanding the knowledge of DNA in biology, so there is a lot of problem. There is a lot of problems. So we should understand that the DNA is the language of life. And then, so DNA is a double strand. DNA is double stranded. It's made of 2 strands. So that is why we call it DNA is a double-stranded. So generally these base pairs, they pair with one another. So there is a pairing. So one nucleotide pair with another nucleotide to form a double-stranded DNA. You can have G, C, T, C, G, A. So generally, if you understand this pairing principle, DNA is double-stranded. And generally, in a DNA, guanine always pair with cytosine. But how do they pair? How do they bind? They bind to hydrogen bonds. Therefore, guanine and cytosine pair to 3 hydrogen bonds. So 3 hydrogen bond pair guanine with cytosine. And then also adenine pair with thymine with 2 double bonds. So the pairing between guanine and cytosine is triple bond. Sorry, 3 hydrogen bond. And adenine and thymine is 2 hydrogen bond. That is how DNA is. So we have guanine here, so it will pair with cytosine, and then cytosine here will also pair with guanine, and then thymine here will pair with adenine, 2 hydrogen bond, and then cytosine also at the same time pair with guanine, and then guanine also pair with cytosine, and then adenine pair with thymine. So this is how the DNA affair has a ladder structure. The structure of DNA is double-stranded, and it appears like ladder. So one nucleotide pair with another nucleotide through a hydrogen bond, and then guanine or a nitrogenous basis linked with another nitrogenous basis through what we call phosphodiester bond, as I mentioned earlier, through phosphodiester bond. So this is how the DNA is. It is made up of double-stranded. It's double-stranded, and it is helical in shape. So ladies and gentlemen, this brings us to the end of the lecture of DNA, and we are going to meet in the next video for the lecture on RNA.